Hi guys, this is Akshaya. In this video, we are going to cover about polymerase chain reaction. To start with synopsis, which includes introduction, steps of PCR, clinical applications of PCR, and types of PCR. Introduction. PCR was invented by Carey Mullis. This is an in vitro DNA amplification procedure in which millions of copies of a particular sequence of DNA can be produced within a few hours. Steps of PCR includes mainly denaturation or separation of the DNA strands is done at 95 degrees Celsius and primers are added. For annealing, cooling to 50 degrees Celsius to anneal the primers, and for extension, with the help of Taq polymerase enzyme, the nucleotides are added at 72 degrees Celsius. So these three steps include the first cycle in which 4 strands of DNA are produced, and then these 1, 2, 3, the 3 steps are repeated, which forms 8 strands of DNA. In the third cycle, 16 strands of DNA are formed, and so in each cycle, doubling of the number of strands is being done. As we see in detail, the first step is separation or denaturation, in which the DNA strands are melted by heating at 95 degrees Celsius for 15 seconds to 2 minutes. In priming or annealing, the primers are being annealed by cooling to 50 degrees Celsius for 0.5 to 2 minutes. The primers hybridize with their complementary single-stranded DNA that is produced in the first step. Polymerization or extension is in which the new DNA strands are synthesized by the enzyme Taq polymerase, which is derived from the bacteria Thermus aquaticus. This reaction is allowed to take place at 72 degrees Celsius for 30 seconds in the presence of deoxyribonucleotide triphosphates. And in the step 4, the first 3 steps are being repeated, so the number of DNA strands in each cycle is being doubled. Thus, 20 cycles provide 1 million time amplification, in which each cycle the number is being doubled. This is being usually done by an automated instrument, that is thermal cycler. After the amplification procedure, the DNA hybridization technique or the southern blot analysis with a suitable probe is done, which shows the presence of DNA in the sample tissue. Now, recent modifications in technology in which all the 3 cycles of PCR are performed at 63 degrees Celsius for 60 minutes. So here the temperature is constantly maintained at 63 degrees Celsius. So here denaturation is not done by higher temperature but with the help of enzymes. This newer technology reduces the cost of the test and the result will be available in a few hours. Next, coming on to the clinical applications of PCR includes diagnosis of bacterial and viral diseases. In the TB sputum, acid-fast bacilli are being amplified by the PCR and then it is being detected by the southern blot analysis. In reverse PCR, the viral infections such as hepatitis C, cytomegalovirus and HIV can be detected after amplification by PCR. Medico-legal cases in which from the crime site, the hair follicles or the blood cells can be taken and it can be for analysis. First, the amplification can be done, which helps in the detection. Next is the diagnosis of genetic disorders, which include sickle cell anemia, beta thalassemia, cystic fibrosis and all. Next is prenatal diagnosis of inherited diseases, in which the fetal cells are being obtained by amniocentesis. Next is cancer detection, in which monitoring of residual abnormal cells present in treated patients can be identified, and identification of mutations of oncosuppressor genes such as P53, retinoblastoma gene, etc., which helps in the identification of individuals who have more risk of cancer. Fossil studies that are used to study the evolution by comparing the sequence of extant and living organisms, and detection of sexually transmitted diseases. Next, we are going to talk about the types of PCR, which includes reverse transcriptase PCR or RT-PCR, in which instead of Taq polymerase enzyme, teeth polymerase enzyme is being used, which this teeth polymerase is derived from Thermus thermophilus, whereas Taq polymerase is derived from Thermus aquaticus. So here, this enzyme, teeth polymerase, has both DNA polymerase as well as the reverse transcriptase activities at higher temperature, which allows the synthesis of both the complementary DNA synthesis and the mRNA followed by PCR amplification. So, viral RNA, that is HIV RNA in the blood, can be detected as early as 4 weeks after infection. Next is nested PCR. After this, we have nested PCR, real-time PCR, quantitative PCR, quantitative real-time PCR, race PCR and multiplex PCR. So in these types of PCR, mainly just one point can be mentioned, or at least the names can be mentioned in the exam. So nested PCR, 2 sets of primers are used in 2 successive PCR runs. The second set is intended to amplify the second target within the first run product. Real-time PCR is used in the quantification of viral load. Quantitative PCR quantifies the DNA or the RNA. And quantitative real-time PCR uses fluorescent dyes such as SERP green or fluorophobe dyes, which measures the amplified product in real time. Next, coming on to race PCR, that is rapid amplification of cDNA ends, that is complementary DNA ends. This technique is used to amplify the ends of the messenger RNA. It is also called as one-sided PCR or anchored PCR. Next is the multiplex PCR, which uses the multiple unique primer sets within a single PCR reaction to produce amplificons of different sizes specific to the different DNA sequences. This is used in the diagnosis of infectious diseases such as viral infections, cancer diagnosis, mutation analysis, pharmacogenomics, chromosomal analysis for cytogenic studies, bacterial genotyping, gene expression patterns in a disease states. So when we write this essay, it is important to mainly write about the steps and the clinical applications of PCR. Other types of PCRs and all, mentioning the name is sufficient. So all the best guys, prepare well and you'll do well in your exams.